So I'm Carson. I'm a postdoc here at Janelia, and I'm going to talk about, oh, sorry, I introduced myself.
You're doing a great job. So we have local talent. Carson will talk to us about high precision coding visual systems.
Okay. So if you're an animal in the world, if you're a squirrel, a monkey, or us, you jump from branch to branch, and you're trying to figure out how to explore the world without falling and hurting yourself. And to do that, you need to have vision in order to not fall, basically. But there's a problem with the brain. You can think of there being a problem with the brain because every single neuron in the brain is noisy. So in the case of mouse visual cortex, we show the same stimulus twice. So I'm going to have this in the video here. So repeat 1 is a set of stimuli, and then repeat 2 is the same stimuli shown 5 minutes later. And if you look at the neural activity, each of these neurons has different responses, even though the exact same stimulus is shown. So you can watch here, and maybe you can look for different neurons. Some of them will have more reliable responses than others. Maybe this one's kind of a decent one down here. But on average, you can see that a lot of the neurons have pretty variable responses from trial to trial. So this might cause a problem in terms of coding the world around you. You want an accurate representation of the world, but every time you see the same stimulus, your responses are different. So we're going to try to figure out if this noise is a problem for coding or if we can basically ignore it. So to answer this question, we do large-scale neural recordings using the mesoscope that Andreas described, built by Nick Sofroniev and Dan Flickinger. And we get around 20,000 neurons on average per recording at 3 hertz recording speed. And then we show visual stimuli to the mice on a screen like this. And here's the mouse that's head fixed, and it's on this large ball, and it's free to run whenever it wants to. So just to summarize the video that I showed, here's an example of a single neuron in response to various different gratings. And you can see each of these dots is a response. And you can see that the responses, even if the grading is, so gratings at 300 can drive a variety of responses, and all of these different, there's quite a lot of variability around this average tuning curve that we fit. So there are some good neurons in the brain that you could think the brain might use. So here's the best neuron we found out of 20,000 neurons that we recorded. It has an SNR of 4, but this is not the norm. So on average, the SNR of these neurons is around this neuron, not this neuron. So maybe this is fine for the brain. The brain has a lot of neurons, so maybe it's OK that they're noisy. We can just basically average the neurons. So we have a stimulus that's 90 degrees, and even if there's some noise, we can average these neurons and get the appropriate angle that the mouse is seeing.
Those are full field stimuli, right?
Yeah, these are full field. All right, so what I was saying basically is most types of noise actually don't limit information. So this independent single neuron noise, we can be fine. We can average the responses and get the correct response to what orientation this branch is at. There's other noise that doesn't matter, and this can also be correlated noise, but in a different space. So we found in previous work that spontaneous behaviors drive correlated activity in the brain, and this activity is actually orthogonal to the stimulus space. So I'll show that to you next, so that you know what I'm talking about. And also there's 1D gain modulation of neurons by arousal, and this noise also doesn't necessarily limit information. So quickly I'll show you this correlated activity that is orthogonal to stimulus activity that will not impair coding because it's not in the space of the stimuli. So if we record the neural activity of a mouse, so here's the neural activity here, and here's the mouse's face. So we're simultaneously looking at the behavior of the mouse along with the neural activity. And here, what's plotted are the principal components, basically, of the motion of the face. And if we look at this video, what you can see is as the mouse behaves, this is in the dark, by the way. This is visual cortex. So there's lots of neural activity in visual cortex, even in the dark, that's driven by the mouse's behavior. And we can show, so here's an example of a recording that Nick Simons did with neuropixels. That's activity all over the brain. And we can predict this neural activity using the spontaneous behaviors, using these face PCs, basically. And so there's a lot of neural activity that's driven by behaviors. And as long as this neural activity that's driven by behaviors is in a different space of the stimuli, it's not going to impair our stimulus coding. And so the next thing we showed in this paper was that the neural activity is orthogonal to the spontaneous behaviors drive neural activity that's orthogonal to the stimulus space. So it's just ongoing activity, and then the stimuli come in and they drive a different subspace. So here's a nice graphical representation of this. So here's neural activity by time, and then these are flashed images. So we flash images during this period of the recording, and then we stop flashing the images during this period of the recording. So what you can see is that, so these are the more behaviorally driven neurons. So we've done this sorting by a manifold embedding algorithm that we've developed. And basically what comes out of this manifold embedding sorting is that the most behaviorally driven neurons are put at the top, and the most stimulus-driven neurons are put at the bottom. And then when the stimuli stop, these directions of stimulus-driven activity, so these directions in space kind of that make up these neurons down here, are no longer driven by the behaviors. They're in a different space of neural activity. And this is just a graphical representation. So if you look at the paper, we have more quantitative analyses of this orthogonality of these two spaces that you can look into. But kind of the point is this ongoing activity, which people have looked at for a long time. They've looked at correlative variability. If it's orthogonal to the subspace of stimulus-driven activity, then it's not going to impair your coding. And that's what we see, is that this at least this behaviorally driven component is not in the same subspace. So to summarize what I just went through, so we have each of our neurons R has some response to a trial T, and then an orientation theta. So say this is responses to oriented gratings, and its response is relative. It's tuning curve F times a gain, which changes from trial to trial, plus this correlative variability from behaviors, which is in an orthogonal subspace. And then the question we're asking in this paper is what's left over. So this model only explains around 40% of the variability. So what we asked in this study is if any of the remaining noise is actually in the stimulus space. So is it actually limiting the information that the mice have about the stimuli? Okay, so we have this independent. So one hypothesis is that the rest of the noise is not in the stimulus space. It's just independent noise. So we can test that by building an independent decoder. So what we did is we took each neuron and we fit a tuning curve to it. So here are 3 example neurons. And then this X is an example trial I'm showing. So these tuning curves are going to give us a likelihood of what stimulus is being shown on that given trial. So in the case of this top tuning curve, we got a high response on this trial. And the average tuning curve says that means that the stimulus is probably this stimulus here. In this case, we got a kind of a low response on this given trial, but this neuron's responses on average are maybe a little higher around here, so it kind of predicts that the stimulus might not be this stimulus, just from this tuning curve. Does that kind of make sense, this likelihood? And then in the bottom neuron, the response to this stimulus, to this trial is high, and it's also, this is the place on the tuning curve where the responses are also supposed to be high, so the likelihood that the stimulus is high. So we're going to basically, we fit a model to each of these neurons, and we find the likelihood that each trial of each stimulus given these neurons, and then we average over all of the neurons, and then we get a prediction of the average log likelihood and a prediction for what stimulus it might be given all of the neural responses. And this is just averaged over all neurons. There's nothing taken into account across neurons. And if we do this, then we get a prediction as follows. So if we look at the true angle that we showed the mice, and then the decoded angle from the neural activity from these 20,000 neurons average, you can see that they mostly fall on this unity line. So it's not a bad prediction. On average, the prediction is around 2 degrees error. And this is across. So this is a histogram of the errors. Basically, if I push this back down and took the absolute value of this line, then I would get on average 2.28 error. And then this is the average across recordings. So on average, if we just use an independent decoder, we can get 2 degrees error. So this isn't too bad, but we still don't know if some of the remaining error that we didn't explain is correlated. So maybe we didn't do a good job because there's actually stimulus correlations that we didn't account for. So what we can do is we can take these errors that we get and see if they're correlated across the population. So to do this, we basically split our neurons into 2 populations. So we have 10,000 neurons in one population, 10,000 neurons in another population, and we do this in a smart way in stripes so that the neurons are never on top of each other in the recording, so there's no bleed through. That's for calcium imaging aficionados. But anyway, so we've split these in a smart way so that we know that there's no bleed through of the signals, and what we see is this log likelihood is very similar for these 2 populations. And if you zoom in, these log likelihoods are similar for both populations, so the error is actually correlated. So if you look at these 2 different populations of neurons, they're telling you that the stimulus is actually not the same as the true stimulus, and these errors are correlated, and I can summarize that basically if the error is high from one population, then it's likely high from the other population. So this independent decoder hasn't taken into account this potential correlations between neurons, and this actually shows that this isn't necessarily the best model. There are actually correlations in the stimulus space that haven't been taken into account by this model. Okay. So these correlations, though, might not be a problem. So first I'm going to say, okay, we're going to rule out the independent single neuron noise model. There are some correlations in the stimulus space. But there's still two hypotheses that remain. So there could be correlations in the stimulus space, but that don't overwhelm the whole population, that we can still decode fine from. So here's an example. So most of our neurons or some of our neurons kind of have independent noise, and then some of them have correlated noise. So there's this one bad neuron, for instance, that's always telling the population it's 100 degrees, and it has this group of neurons it's always influencing. And then maybe there's another bad neuron that's always influencing another group of neurons. But if it's not the whole population that's influenced, we can actually just get rid of these neurons in some sense and get the correct answer. And kind of getting rid of correlations in the population is exactly what you do when you do linear regression. You kind of take into account the fact that neurons are correlated to each other and kind of whiten the activity, and then you get the correct responses. So maybe we can just use a linear decoder and actually get the correct response. There's another hypothesis, though, which is that actually that more of these neurons are actually correlated to each other. So this was proposed by Ruben Moreno-Botti. So these are correlations that actually will be problematic for the brain. So what happens here is that the whole population shifts its activity. So all the correlations in the population, the correlations are across the whole population, and at any given stimulus, all the neurons are influenced to tell the brain, or basically in the brain, and say that the stimulus is different. Okay. You might think this is kind of a crazy thing to think that this might happen, but so we're going to try to distinguish between the two. But I'm just going to tell you some examples of where this information-limiting correlations might arise. So one hypothesis is that the noise is coming before visual cortex. So say there's noise in the retina or in the thalamus, then basically your orientation, if the thalamus, the on-off cells or whatever are biased in some way, then this information is lost before it even gets to visual cortex, and all the neurons are going to think the stimulus is wrong. So that's not a totally crazy thing to think might happen. Another hypothesis that would predict information-limiting noise is that there's an attractor in visual cortex. So if, for instance, our orientations lied in some kind of attractor regime, so like anytime a little bit of noise came into the system, it perturbed the whole population to think the angle was slightly different. So that's what, in an attractor, basically, you're stuck in this kind of ring, and any noise is just going to perturb you along this ring. It's not going to perturb you in different directions. So you'd be in trouble because the whole population is going to have perturbed different activity. So if we were in a attractor state, you could also get these information-limiting correlations. All right, so let's see if these information-limiting correlations actually exist. So we're going to see if we can take care of these correlations in the stimulus space with a linear decoder. So we're going to use linear regression, and we're going to do a regularized linear regression. So we're basically going to summarize these neurons by superneurons. So we take like 30 of these superneurons, which just have these nice von Mises tuning curves at different orientations, and we're just going to try to predict these superneurons using 75% of the data. And then on the test, we can see what the responses of these superneurons look like. And then for a given trial, so here's a response to a given trial. Here, this white dot. We can take these responses and predict the angle based on what the preferred angle of this von Mises is. So in the case of this stimulus, sorry, this is kind of dark, but you see this kind of black tuning curve. The responses are higher for this von Mises. So we're going to say that the angle is around, I don't remember what this one is. Maybe it's around 200, yeah. So the angle is going to be around 200 because this von Mises has the highest response. So that's how we're going to decode the stimulus. So we have these basis functions, basically, and we're going to say whatever basis function has the maximal response, that's the angle that is the decoded angle. So we can kind of project these dots onto this line and then get the decoded angle is somewhere in between these two von Mises functions. And then we get the decoded angle is 200, which the original angle, I think, was 201. So this is good. This is closer. So if we look across test trials, if we look at the true angle versus the decoded angle for a linear decoder, we're even more on the line. And so we're doing a better job of predicting by taking into account these correlations. And if I show you the histogram again, the error is around 1 degree. And the median error across recordings is also around 1 degree for all of these different recordings. Okay. So we're at 1 degree error, but there's a possibility that we could even go even lower. So we don't know if this is a limit, if there's information limiting correlations remaining, or if we can reduce this information even lower if we increase the number of neurons and the number of trials. So if we looked next at the asymptotic performance of this decoder. So if we increase the number of neurons, this is the decoding error. So the bottom point here is this 1 degree error that I was talking about. And we fit these functions to these curves to approximate how much the variance should scale as you increase the number of neurons or you increase the number of trials. And we found that if you fit these functions of number of neurons and number of trials, then alpha, which is kind of this asymptotic line, it's a bit lower if you increase the number of neurons, but it's even lower if you increase the number of trials. So it's predicting that if we had more trials, maybe we'd asymptote closer to 0.3 degrees. And we can actually, so we can't increase the number of neurons, and the brain can't either in some sense, but we can increase the number of trials. And you might think we only showed, so we did 5,000, we showed 5,000 stimuli to 360 angles. So maybe we didn't have enough trials to fully accurately fit this model, and maybe we were kind of overfitting. So the simplest thing to do is just increase the number of trials to, all right, I'll show the slide, sorry. So increase the number of trials within each degree. So instead of showing 4,000 or 5,000 trials to 360 different degrees, we'll show 4,000 trials just within 4 degrees. So we'll just show within 43 degrees and 47 degrees. So this is like almost 1,000 trials per degree we showed the mouse. And there might be some more adaptation and so forth, but we actually were able to decode from this. And we changed the task a little bit. So now we're no longer decoding from 360 degrees. Now we're just looking at, is the response, for instance, so we show between 43 and 47, and we're just asking on every single trial, is the angle greater than or less than 45 degrees? And so if we look at the neural responses and we see how accurate they are, basically how likely are they to say 44 or 46, or how likely they are to get this problem right, they get the problem right 75% of the time at 0.3 degrees. So what this is saying is that there's a lot of information in visual cortex, and if you have enough trials to build the right model in some sense, you can really decode very well from these stimuli what the true stimulus is in the world within 0.3 degrees. And just to give you some context, humans can perform within about 1 degree once they're trained. If you're an untrained human, you're not going to do quite so well. Like maybe you can look, I could have hidden these, but maybe you might have a hard time doing this discrimination, untrained. But people have also trained mice to do this task. And mice, so this is a totally different axis you can see. The mice do much worse on this task than humans and their brains do. So there are so now we wanted to ask, what is this discrepancy? So so first I'll summarize, the neural activity is very precise. We don't really see any evidence of information limiting correlations. If you go to the bioRxiv preprint, you can see more asymptotic analyses to kind of nail down this point. But so there's a lot of information in visual cortex, but it's not necessarily being used by the mouse when it's doing a behavioral discrimination task. So then we asked, why is the behavior so much worse than the neurons? So one hypothesis is that these brain states in the brain are modulating decoding. But what we found in the paper, you can look it up, is that there's only slightly worse decoding in passive states versus running states. So we have periods where the mouse runs and periods where the mouse is passive, and we didn't see huge differences in the amount of the discrimination threshold, so how well we could predict the neural activity. So it's not necessarily the brain states, at least in the case of a mouse just sitting there running or not running. Another hypothesis is that maybe this information is locked away in V1. So maybe it doesn't actually leave V1, so the mouse doesn't have this information to use in downstream decoders. So what we did is we did recordings in higher order visual areas and used the same task where we in some sense. We showed images between 43 and 47 degrees, and we got the same performance of 0.3 degrees. So this information does at least leave V1 and get to higher order visual areas. I mean, the question is where it goes from there, but that's obviously for future work. So one other hypothesis is that the problem with the behavior is that people have shown lots of different stimuli when they when they do behavioral tasks. So sometimes they show really short stimuli, sometimes they show gradings that are sign gradings or random phase gradings. So in our case, sorry I didn't say this, we always use square gradings. And sometimes they show small stimuli. Sometimes, I don't think they show Minnie Mouse, but we showed Minnie Mouse and we got similar performance. So, and they show drifting gradings and low contrast and noise. And what we found is that there's similar accuracy in the brain to all these stimuli. So that's not it's not necessarily that people were using different behavioral paradigms that was the problem, in different stimulus paradigms in their behaviors. That was the problem. So this isn't necessarily the reason why the behavior is different. Okay. One other hypothesis is that the brain doesn't have instantaneous access to all the training trials. So when I built my linear decoder, I did linear regression on all the trials that I had from the responses. But the mouse, if you think of a mouse doing a behavior, it doesn't necessarily have access to all of that. It might be doing some sort of reinforcement learning where it's constantly updating its prediction of what oh, sorry, I'm out of time. Okay, anyway, what we showed in the paper is that incremental learning with perceptrons also works well. So if you can use incremental learning and linear perceptrons to do this behavioral task. So that's not the problem either. So in summary, we found that a linear decoder beats an independent decoder. So some of the correlations in visual cortex are detrimental, but we can take care of them by using a linear decoder. So we found the actual error was close to 0.3 degrees, and the asymptotic estimate is around 0.1. So these correlations are not information limiting. Even if they can be detrimental if you just use an independent decoder. And then we don't know what this means for behavior. We haven't found we haven't really found the hypothesis for why the behavior is worse, and that's for all of us to try to figure out. So thanks to my advisor, Marius Pakitsariou, and Mihaly, he did all of the recordings that I showed you, and we have technical support from people at Jamelia. That's really helpful. And the paper's on bioRxiv, the data's on Figshare, and the code is on GitHub. And so we have 37 data sets, so around like 700,000 neurons. So have fun, theorists.
I was wondering whether we would see more accuracy in behavior if you were to do a change detection task.
Yeah, and people do see that. So we didn't actually do the behavioral discrimination ourselves, but yeah, so people do see more accuracy in change detection tasks, but that's also an easier problem for the brain to solve because you just need to look if one pixel changes. This is a harder task in some sense to solve, I would say. So, you can say something else, sorry.
I wanted to say that depending on how you do the change detection task, rather than presenting an image and then the next, you can present an image, then a gray screen for a few milliseconds, and then the next image, and then anyway.
And then it becomes more of a memory task in some sense.
And then you can shift the images.
Sorry, maybe you kind of said that in a different way, but one of the questions that I always had about trying to do this neurometric, like relate how much information the neurons encode, especially with this type of stimuli versus the animal, is that the animal doesn't, is trying to reconstruct the stimulus, right? So it just has activity. It doesn't know that the stimulus is an orientation grading. It's trying to infer that stimulus itself. So, so if you just if I don't tell you what the experiment is that you're doing, that's kind of what the animal is trying to reconstruct. So it may not be that surprising that there's a big discrepancy between neurometric functions and behavior because the brain is trying to do a lot of other things. Right? Have you thought about that or how you can address this experimentally?
Yeah, I definitely agree with you. I think it's that these tasks are not ethological, and that's the reason the mice might not necessarily be able to do them. I think, and kind of the point with this study, hopefully, is that people see that mice can actually see very well, and they can see high frequencies like you've shown as well. And so we just need to figure out the right tasks that are ethological, that they're used to doing. Like maybe, navigation, for instance, is something they're very good at, and other sorts of things.
Is your data set, oh, sorry, Corral. I can, yeah, but you're.
Carson, I must have asked this before. I still don't understand why you. I mean, this is the for the first part. What does that mean? Saying that the behavior drives the activity. I don't really understand that. I mean, there are obviously a lot of people who worry about reafferents, efferents copies, and so on and so forth. And without detailed analysis of loops, it's hard to know what drives what, right? I mean, is that right, or am I missing something?
Yeah, I agree with you. So in some brain areas, so you can look at the coincidence of the behavior with the neural activity and the neuropixels data. You can't look at that in the calcium imaging. And some of the brain areas have shorter lags than others. So it could be simultaneous with the behavior because of afferents from motor cortex or something. Or it could be feedback proprioception. And it's not clear what either of those are. So yeah,
I'm not sure how the neuropixels work. I mean, we've done this thing in barrel cortex a lot, and with whole cell and loose seal and millisecond time scale, it still is always very hard. And you know, you have to cut sensory nerves, you have to cut descending inputs to really kind of make statements about who drives who, right? I mean, obviously, neural activity, you know, the brain is just a couple of synapses from the motor system. It would be very, very surprising if neural activity in the brain is not reflected in some sense in movement, right? I mean, that's, yeah. So.
No, I agree with you. I don't think it answers that question at all, but at least says that it could go either way, and it could be any of those mechanisms. It doesn't rule out any of them. Maybe I shouldn't have said it in such a controversial way on my slide. Yeah.
So there is another factor that will influence decoding accuracy is the duration of the interval that you're using for computing your for decoding. So I was wondering, first, what's the duration of the interval that you're using, and if the brain uses a much shorter temporal interval, then that might explain part of the discrepancy.
Yeah, we thought about that too. So we always showed about three. We always were using around 2 or 3 time bins, which is around 700 to 800 milliseconds or to a second. And we also showed short stimuli, and we saw similar decoding accuracy. But we don't. I mean, I think that's a question that's going to have to be answered fully with EFIS. But the fact that you can decode short stimuli is a sign that at least that information is there, even if the stimulus is brief. But maybe we need more neural activity to average and get the right response.
One question. It's a general question, but again, related to the behavior discrepancy with the decoding. For anybody in the room, has anyone shown that layer 2-3 is required for any of these tasks? I mean, it would be weird, given how well it does from a decoding perspective, but it's also possible that there are other neurons you're not imaging that are primarily involved in the behavior.
I think for orientation discrimination, you need visual cortex.
Not visual cortex, layer 2-3.
Layer 2-3, oh, no. But I mean, we see similar decoding performance in layer 4, so it could use layer 4 as well. But yeah, I mean, it's anyone's guess what the mouse is using. Yeah.
All right, let's thank Carson.